If you've been wondering why your skin looks duller than it did 2 years ago, why a small workout leaves you sore for 4 days, why your memory keeps slipping on names you've known forever, and why you wake up feeling like you didn't actually sleep, I want you to stop blaming the calendar, stop blaming your parents' genes, stop blaming the slow march of time, because there is something happening inside your body right now, deep inside the marrow of your bones and the tissue of your brain, that almost no general practitioner will mention during a 15-minute checkup. The story you've been sold is that stress is a psychological issue, something you fix with a meditation app, a long weekend, or a glass of wine after work. But the laboratory data tells a very different story, and once you see it, you can't unsee it. Chronic stress is not just an emotional state. It is a biochemical event that quietly disassembles the exact system your body uses to rebuild itself every single day. And there is one plant used by healers in northern India for more than 3,000 years that modern, peer-reviewed research has now shown acts on both ends of this problem at the same time. Stick with me, because the next several minutes may permanently change the way you understand fatigue, recovery, and aging itself. Most of us were raised to think stress lives in the head, that if we just slow our breathing or take a hot bath, the body resets on its own. But in nearly a decade of working in emergency rooms and then transitioning into longevity-focused medicine, I started noticing something that completely rewired my thinking. Stress is not abstract. It is measurable. It leaves fingerprints inside your cells long after the argument, the deadline, or the difficult month has ended. And the molecule that carries those fingerprints is cortisol. When cortisol stays high, even at levels your doctor might call normal, it doesn't just make you feel wired and tired. It actively shuts down the production and movement of your stem cells, which are the workforce your body depends on to patch itself back together. That is not poetry. That is documented in published literature, and most importantly, it is reversible. My name is Dr. Leana. I work in emergency medicine and I research integrative approaches to longevity. And I want to be honest with you about something. For the first several years of my career, I treated traditional plant medicine the way most of my colleagues did, with a kind of polite dismissal. If a compound hadn't been through a double-blind trial, I didn't take it seriously. Then, a colleague handed me a stack of papers on Withania somnifera, the plant most of the world knows as Ashwagandha, and what I read was not folklore. It was rigorous, mechanism-level, dose-controlled pharmacology showing that this single root does measurable things to human physiology that we are only beginning to map in detail. It acts directly on the same biological systems I had been watching collapse in my chronically stressed patients. The stem cell environment, the HPA axis that governs cortisol, and the smoldering inflammation that quietly ages every tissue in the body. That shift changed my practice, and today I want to walk you through what I learned. I'm going to cover 5 things that almost nobody, including most physicians, knows about this plant and your biology. First, how sustained cortisol elevation quietly destroys your stem cell reserves, and why this is the real engine behind premature aging. Second, the specific molecule inside ashwagandha that has been shown in lab settings to wake up dormant neural stem cells. Third, the 4 separate biological pathways this root works through at the same time, and why that combination matters far more than any single mechanism. Fourth, the exact protocol grounded in clinical research, which form to choose, what dose to take, when to take it, and which foods to combine it with for amplified effect. And fifth, the 3 avoidable errors that cause most people to get nothing out of ashwagandha, and the simple corrections that fix each one. Quick favor before we dive in. If you've been watching my videos for a while and never tapped subscribe, please do it now. It's completely free and it genuinely helps this channel grow. You can also follow me on Instagram, where I'm still very small, and everything I reference in this video is linked below so you don't have to hunt for it. Now let's talk about stem cells, because most people misunderstand what they actually are. When the average person hears the term, their mind goes to embryos, controversial therapies, or expensive treatments offered overseas. But you have billions of stem cells living inside your body at this very moment, in your marrow, in your brain, in the lining of your intestines, in your muscles, in the walls of your blood vessels. Picture them as a specialized repair crew, permanently stationed throughout your tissues, waiting for a dispatch signal. When a blood vessel cracks, when a neuron starts to die, when a muscle fiber tears during a workout, these cells receive the call, migrate to the damaged area, and rebuild what was lost. This is not a metaphor. This is happening inside you every second of every day. Now, here is where cortisol enters and where the story turns serious. Cortisol is your body's main stress hormone, and in short, sharp bursts, it is genuinely useful. It sharpens your senses, pulls blood toward your muscles, and prepares you to act. That is exactly what it evolved to do. But imagine your repair crew has a foreman, and imagine that when cortisol stays high for weeks, months, or years on end, that foreman starts giving a very specific instruction to every cell on the team. Hold position, conserve energy, do not rebuild. That is essentially what happens inside your tissues during chronic stress. Research published through the National Institutes of Health has confirmed that glucocorticoids, the hormone family cortisol belongs to, directly slow stem cell division in bone marrow, reduce neural stem cell activity in the hippocampus, and lower the regenerative output of the cells that line your gut. The crew is still there, but it has stopped responding to damage. And meanwhile, every single day, tiny tears, leaks, and breakdowns pile up without being repaired. Small leaks become structural failure, and over years, that failure shows up as cognitive fog, weakened immunity, metabolic disorder, and a body that simply looks and feels older than it should. But here is the part that matters most. None of this is fixed in stone. None of it is dictated by your DNA. It is a response to a hormonal message, which means changing the message changes the result. That is precisely where ashwagandha enters the picture. The plant has been documented in Ayurvedic manuscripts for more than 3 millennia, and for most of the history of Western medicine, that long tradition was treated as a reason to ignore it. But what has happened over the past 20 years of laboratory work is genuinely remarkable. Modern pharmacology did not discover something new about this root. It confirmed what traditional healers had been saying for centuries, and then explained the molecular reasons behind their observations. The root contains a class of naturally produced steroidal lactones called withanolides, and when scientists isolated these compounds and tested them on human cell lines and animal models, the results were not subtle. One compound in particular, withanolide A, the most studied of the group, demonstrated direct neurogenic activity. It crossed the blood-brain barrier. It engaged stem cell signaling pathways inside the brain. And it did something that almost no pharmaceutical molecule has been intentionally engineered to do. It appeared to simultaneously lower the stress hormone that suppresses stem cells while directly encouraging the growth of new ones. Two opposing problems addressed by a single root. Let me walk you through how, mechanism by mechanism. The first pathway is direct activation of neural stem cells through withanolide A. In a study printed in the British Journal of Pharmacology, researchers showed that withanolide A, isolated from ashwagandha root, promoted the proliferation and maturation of neural stem cells in the hippocampal region of the brain. The hippocampus is the seat of memory and learning, and in chronically stressed individuals, it is one of the first structures to physically shrink. What withanolide A appears to do is essentially flip the grow switch back on inside progenitor cells that go dormant under stress. This is not theory. It was observed under direct laboratory conditions with cell culture models, where colony expansion can be measured in real time. The second pathway is suppression of the HPA axis, which means turning off the cortisol faucet at the source. The hypothalamic pituitary adrenal axis is the command chain that orders cortisol production. Under chronic stress, this system behaves like a throttle stuck halfway open, continuing to release cortisol long after the original trigger is gone. Ashwagandha's withanolides appear to recalibrate the sensitivity of this loop, supporting the feedback mechanism that tells your body cortisol levels are already high enough. Multiple clinical trials have measured this effect, not through questionnaires about how stressed people feel, but through actual serum cortisol assays drawn from the bloodstream. When cortisol falls, the suppression signal aimed at your stem cells lifts, the foreman stops ordering the crew to stand down, and repair resumes. The third pathway, often overlooked, is the restoration of anabolic hormones. Testosterone and growth hormone are not just markers of vitality or athletic performance. They are direct activators of satellite cells, the stem cells that rebuild muscle and bone. When cortisol stays elevated, testosterone declines because the body in survival mode chooses defense over construction. A trial published in the journal Fertility and Sterility involving 75 men under chronic stress found that those taking 300 milligrams of standardized ashwagandha root extract daily showed a statistically significant rise in testosterone and a 167% improvement in sperm count over 90 days. Higher testosterone directly reactivates the muscle and bone stem cell populations that cortisol had been silencing, and this is what I call the anabolic restoration effect. The fourth pathway is suppression of NF-kappa B, which clears the inflammatory haze that surrounds dormant stem cells. Stem cells do not exist in isolation. They live inside a microenvironment called the stem cell niche, and the condition of that niche determines whether they activate or stay quiet. Chronic inflammation driven by NF-kappa B creates an environment around stem cells that is essentially toxic. Imagine a construction team trying to rebuild a road while a brush fire burns around them. The fire does not destroy the workers, but it makes it impossible for them to do anything productive. Ashwagandha's compounds are documented inhibitors of NF-kappa B, calming the inflammatory signal and clearing the air so stem cells can finally get back to work. This anti-inflammatory action works hand in hand with the cortisol-lowering effect because together they remove the two biggest obstacles standing between your body and its own repair capacity. Now, let me anchor all of this to specific human studies. A randomized, double-blind, placebo-controlled trial published in the Indian Journal of Psychological Medicine followed 64 adults with chronic stress. Participants given 300 milligrams of high-concentration ashwagandha root extract twice daily for 60 days experienced a 27.9% reduction in serum cortisol compared to placebo, measured through laboratory cortisol assays, not self-reporting. A second study from the National Brain Research Center in India examined the effect of withanolide A on neurons in animals with damaged nervous tissue, and the compound not only encouraged regrowth of axons and dendrites, the wiring of nerve cells, but also stimulated activity in neuroprogenitor cells, which are essentially the brain's version of stem cells. This was the first time a naturally occurring compound demonstrated this kind of regenerative effect in a controlled living model. A third trial, published in 2019 in the Journal of the International Society of Sports Nutrition, measured the effect of 600 milligrams of KSM-66 ashwagandha daily on muscle recovery and anabolic hormones across 40 healthy adults over 8 weeks. Participants experienced a 15.2% greater rise in testosterone, significantly improved recovery measured through creatine kinase markers, and meaningful gains in VO2 max. Faster recovery is the direct downstream signature of muscle stem cells doing what they were built to do. A fourth study, published in the journal Medicine, followed 58 people with non-restorative sleep, and those taking ashwagandha root extract showed a 72% improvement in sleep quality scores and a notable drop in morning cortisol, meaning the nighttime calming effect carried into the following day's hormonal environment. This matters because deep sleep is when the majority of stem cell repair work actually happens, so improved sleep multiplies your repair window. Now, let me give you the protocol exactly the way I outline it for patients. First, choose the right form, and this part is not flexible. Not every ashwagandha product is equivalent. The two extracts that have been clinically validated are KSM-66 and Sensoril. KSM-66 uses only the root and is standardized to at least 5% withanolides, and it is the most studied form for testosterone, cortisol, and physical performance. Sensoril uses both root and leaf and has stronger evidence for cognitive function and sleep. If your main targets are cortisol reduction and stem cell regeneration, start with KSM-66. Avoid the generic ashwagandha powder sold without a standardization percentage on the label because you genuinely have no idea what is inside it. Second, dose. The clinical sweet spot is 300 milligrams twice daily, 1 in the morning with food and 1 in the evening before bed, for a total of 600 milligrams. If you are new to it, start with 300 milligrams once a day for the first 2 weeks to gauge how your body responds. Third, Take it with fat. Withanolides are fat-soluble compounds, so taking your dose alongside a meal that includes avocado, olive oil, or nuts substantially improves absorption. Do not take it on an empty stomach. Fourth, the evening dose is the most important. Your body performs most of its repair work during deep sleep, so taking ashwagandha 30 to 60 minutes before bed both improves sleep quality and aligns the cortisol-lowering effect with the hours when it has the biggest impact. Fifth, stack it with foods that work synergistically. A cup of blueberries supplies polyphenols that support neural stem cell activity. Turmeric paired with black pepper amplifies NF-kappa B suppression, and the black pepper increases curcumin absorption by roughly 2,000%. 30 grams of pumpkin seeds daily provides one of the richest plant sources of zinc, which is essential for testosterone production and stem cell signaling. Fatty fish or ground flaxseed delivers omega-3s that support the anti-inflammatory niche around your stem cells. And about 30 grams of dark chocolate at 70% cacao or higher supplies magnesium, which supports cortisol regulation overnight. Sixth, cycle the herb. 8 weeks on, then 2 to 4 weeks off. This is not optional. Adaptogens like ashwagandha operate through hormonal feedback systems, and running them continuously can dull that responsiveness over time, so cycling preserves sensitivity and keeps the effect strong. After years of working with patients on cortisol-related issues, here are the clinical observations I find most people miss. First, test. Do not guess. Before you begin, ask your doctor for a simple morning cortisol blood test. 4 to 6 weeks into the protocol, test again. If your number is dropping, you have biological proof the protocol is doing its job, not just a vague sense of feeling better. Second, pair it with brief cold exposure. A 2 to 3 minute cold shower in the morning produces a sharp, controlled cortisol spike, and this kind of acute hormetic stress actually trains the HPA axis to recover more efficiently afterward. Combined with ashwagandha's calming effect in the evening, the contrast between morning and night creates a regulating rhythm across your entire cortisol cycle. Third, magnesium amplifies everything. Magnesium deficiency, present in roughly 60 to 70% of adults, directly disrupts cortisol regulation and undermines sleep architecture. Taking 200 to 400 milligrams of magnesium glycinate at night alongside your ashwagandha dose removes one of the biggest barriers standing between the herb and its full effect. Now I want to address the 3 mistakes I see constantly, because they explain why so many people try ashwagandha for a few weeks, feel nothing, and give up convinced it's hype. The first mistake is buying unstandardized powder. Most of what fills the shelves at grocery stores and budget supplement aisles is raw ashwagandha powder with no withanolide standardization printed anywhere on the label. The active compounds are technically present, but the concentration could be anywhere from 0.5% to 8%, and you have no way to know. The fix is simple. Look for KSM 66 or Sensoril clearly named on the bottle, and look for the phrase standardized to a specific percentage of withanolides. If neither appears, put it back and walk away. The second mistake is taking it only in the morning. Most people swallow all their supplements at breakfast out of habit, but ashwagandha's most valuable effects, cortisol regulation and deeper sleep, are unlocked specifically by the evening dose. The fix is to split your dose, half with breakfast and half 30 to 60 minutes before bed. The third mistake is expecting results in 2 weeks. This is the single most common reason people quit. They try the herb for 10 or 14 days, notice nothing dramatic, and conclude it doesn't work. But here is the truth. You are attempting to reverse a hormonal pattern that may have taken years to build. Cortisol-driven stem cell suppression unfolds slowly, and reversing it unfolds slowly as well. The trials that produced the most striking results ran for 60 to 90 days, so commit to a minimum of 8 weeks before drawing any conclusion. And during that window, track objective signals like sleep depth, recovery speed, and energy stability, rather than asking yourself each morning whether you feel less stressed. I want to leave you with something important. The idea that your body's ability to heal itself declines automatically with age is partly true, but it is far from the whole story. A large portion of what we label as aging is actually the slow accumulation of years of elevated cortisol, quietly dismantling a repair system that is still entirely capable of functioning. Your stem cells are not gone. Your capacity to generate new neurons has not vanished. Your hormones have not forgotten how to balance themselves. They have simply been silenced by a signal, a measurable and reversible hormonal signal. The people who keep their minds sharp, their bodies resilient, and their immune systems strong well into their 70s and beyond are not biologically gifted or genetically lucky. They have built habits, sometimes deliberately and sometimes by instinct, that keep this internal repair system alive and active. Those same systems exist inside you right now, and an ancient root validated by modern science may be one of the most accessible and powerful tools available to wake them up. You can begin today. Let me pull everything together so you walk away with a clean picture. Chronic cortisol elevation, not occasional stress, but sustained elevation, directly suppresses stem cell activity in the marrow, the brain, and the gut. And this is biological suppression you can measure, not a feeling you can dismiss. Ashwagandha, and specifically its active compound, withanolide A, works through 4 overlapping pathways at the same time, directly activating neural stem cells, modulating the HPA axis to lower cortisol, restoring testosterone and growth hormone to reawaken muscle stem cells, and suppressing NF-kappa B to clear the inflammatory environment around those cells. The research backs this up with a 27.9% measurable cortisol reduction in 60 days. Withanolide A triggered neuroprogenitor growth in controlled studies, a 15.2% rise in testosterone with measurable VO2 max gains over 8 weeks, and a 72% improvement in sleep quality during the very window when stem cell repair is most active. Your protocol is KSM-66 or Sensoril only, 300 to 600 milligrams daily, split between morning and evening, always taken with fat, stacked with blueberries, turmeric, zinc from pumpkin seeds, and magnesium glycinate at night, and cycled 8 weeks on with 2 to 4 weeks off. Avoid unstandardized powder, take the evening dose seriously, and give the protocol 8 to 12 weeks before you judge it. Your health is not written in your genes, and it is not chained to your age. It is shaped daily by what you give your biology to work with, and you have far more influence over the outcome than you have been led to believe. If this video gave you something worth holding on to, please subscribe and tap the like button. It takes 2 seconds, and it genuinely helps this channel reach more people searching for honest, science-grounded answers about their own bodies. I'll see you in the next one. Dr. Leana, signing off.